Thank you so much. That's such a wonderful introduction, Raya. I'm going to start with my disclosure statement. I do get funding from the Chow Foundation, which gave us some pilot funds for the UW Jedi Center, and have had some research funding through the Cooney Foundation. And so I'm going to start with a little bit about the Office of Healthcare Equity, which is my new gig. I've been in the office, now it's going to be 2 years coming up, I think in April. And I am privileged to work with the office. And we just recently went through and came up with a new Blueprint 2.0, which is the path to health justice. And I'll start with a statement honoring the land on which the University of Washington stands. We at UW Medicine acknowledge the land we occupy today as the traditional home of the Tulalip, Muckleshoot, Duwamish, and Suquamish, or Coast Salish tribal nations. And without them, we would not have access to this healing, working, teaching, and learning environment. And we humbly take this opportunity to thank the original caretakers of the land who are still here. And I also want to acknowledge that February is the beginning of Black History Month, so I also want to acknowledge all of those who worked during the building of this country to make it what it is. The team that I work with includes Paula Houston, who is the Chief Diversity Officer and my boss, and Leo Morales, who's Assistant Dean for Quality Improvement, Edwin Lindo, who's the Assistant Dean for Social and Health Justice, Jonathan Cantor, who's a behavioral scientist and leads our training activities, Eric Ho, who's Director of Operations, and Lee Davis, who is our training manager and leads our foundational training. Martine Pierre-Louise is our EDI Director at Harborview, and Elena Casio is our EDI Director at Montlake. This is our team, and we address a lot of the issues that have to do with equity, diversity, and inclusion. So this is our blueprint. We have about 6 objectives, strategic focus areas that include the learning environments, our workforce development, quality improvement, research and evaluation, community engagement, and leadership and strategic operations. So I won't go through everything, but I'll just give you a few highlights of some of that and tell you a little bit about what we're doing. So in regards to leadership and strategic planning, we help and sort of help with a lot of the faculty searches. So we do have a new search out for the Assistant Dean for Equity, Diversity, and Inclusion in Research and Graduate Education. And this person will work with the biomedical research departments and with our office. So that search committee has been formed and the position description has been created and the internal search is starting. I've also been on the Assistant Dean for Equity and Medical Student Engagement search committee, and that search is ongoing, and that's a new position that will work in our undergraduate medical education site. We also do a lot of work with our workforce, so the Committee for Minority Faculty Advancement, some of you may be on this or know about it. We have a new charter that's been approved. The outgoing chair is Johnny Orozco, who's in oncology, and the incoming chair is Hannibal Pearson, who is a gastroenterologist at Children's. So we're very excited about this new partnership. For community engagement, we do a lot of community sessions, especially for our LGBTQIA and other allies. We also have a community engagement that we're starting and have community conversations that are ongoing. We had a lot of community conversations that had to do with COVID, a lot of pop-up clinics. Just in regards to the LGBTQ work that we're doing, we do have someone in our office who works on that. We have an out list for those who are identified as students, trainees, or faculty or staff. And we have a QMed mentorship program and recruitment that's run by MB Vasquez, who's in our office now. In regards to the undergraduate medical education, there's a new pathway called the Black Health Justice Pathway that was started by for medical students, and it's focused on Black health justice. They have the leadership team. You can see those students there: Jasmine Galt, L'Oreal Kennedy, Eileen Lee, and Jeremiah Sims, and it's led by Dr. Peter Asante. And they developed a new course that's led by Dr. Ali Thomas and Patricia Egwatu. So this is an exciting program. We have other pathways in our office: the Native American pathway, Latinx pathway, LGBTQ pathway and the CUSP, which is a community underserved pathway. We also have the WISE Center that is led by Estelle Williams, who's one of our faculty here. She's a surgeon. And these programs are sort of pre-medicine ecosystem programs. So they include our pre-health programs like Doctor for a Day, UDoc, the Summer Health Professions Education Program, and other health sciences programs. And she does a lot of work with all of our communities out there. And the Doctor for a Day is sort of the signature program, but also the Summer Health Professions Education Program, which is a longstanding program that's been supported by the Robert Wood Johnson Foundation and support from this department as well. So I just want to acknowledge that and thank you for all of the support for our programs. And the programs sort of run from elementary school all the way to middle school, high school, and undergraduate, and just are very important for our office. We are also working on restorative justice at the University of Washington and are starting to build restorative justice. Some of you may have taken part in some of the community building exercises, and that's something that we're offering more training in, and wanted to acknowledge that. We're starting with the strategy of educating our UW community about restorative justice practices, sort of in a stepwise phased process. We're going to train, so key leaders will offer training to faculty, staff, and students, and then develop an enduring home for restorative justice here at the University of Washington. So before I continue, I'll just ask any questions about that because I'm going to switch gears and talk more about my research, or we can wait till the end. So thanks. All right, so I'm going to switch gears. I'm going to talk about health inequities, APOL1, chronic kidney disease in African Americans and Blacks. And the objectives of this talk are really to review the causes of health inequities and chronic kidney disease. As I said, I'm a nephrologist, so my research really focuses on kidney disease and African Americans and Blacks. So, I'll also describe how the link between APOL1 or Apolipoprotein L1 or APOL1 was made in kidney disease, and then reflect on the ethics of APOL1 genetic testing in kidney disease. So today is February 1st, Black History Month, so I start my slide with this of all the forms of inequity. Injustice in healthcare is the most shocking and inhumane, and that remains so. And so I just wanted to bring that to everyone's attention. This is Martin Luther King. He said this over probably over 60 years ago, 50 years ago, still true today. And I will start with the case. So this was a patient that I had when I was still seeing patients at the VA. So this is Mrs. J. She's a 56-year-old African-American female. She has a history of diabetes, hypertension, and an elevated creatinine. She has a past medical history of hypertension, diabetes she's had for 10 years. She has a family history of end-stage renal disease or end-stage kidney disease. I'll probably flip back and forth between those two. They mean the same thing, and two relatives. And when I saw her in clinic, her blood pressure was a little bit on the high side. Lungs were clear, cardiovascular, regular rate and rhythm, abdomen was soft and non-tender, and she had some edema. Her creatinine at the time was 2, and her EGFR was 20, and hemoglobin A1C was 8.5, so diabetes was a little elevated, or not as well controlled as it could have been. Her ultrasound showed that she had 2 normal-sized kidneys, or 2 kidneys that were 11 centimeters, so a little bit large, and her biopsy showed that she had diabetic nephropathy and focal segmental glomerular sclerosis. So we expected her to have diabetic nephropathy. Focal segmental glomerular sclerosis, as you all probably know, is higher and found to be higher in African Americans, which is thought to be due to the higher incidence of hypertension. So even though we controlled her blood pressure and we controlled her diabetes, she rapidly progressed to needing dialysis over the next few years. And this was before SGLT2 inhibitors, so we didn't have the benefit of that new medication. She underwent a gastric bypass and lost a significant amount of weight, which helped her diabetes. And then she received a transplant from her 18-year-old daughter and is doing well and has normal kidney function. So one thing to think about are what factors are important in thinking about transplantation, and what are some other tests that are now available that weren't necessarily available back then when we saw her in clinic. So I'm going to talk a little bit more about kidney disease and the background and how it disproportionately affects African Americans. There are about 37 million people who have chronic kidney disease in the U.S., and African Americans and blacks have a 2 to 4-fold greater risk of development of kidney failure or end-stage kidney disease compared to whites. And if you go into a dialysis unit, about 32 to 35% of dialysis patients are African American, but African Americans only comprise about 12% of the population. If you look at the incidence of kidney disease, it's greater in racial and ethnic minorities than whites. So this is data from the United States Renal Data System, which is a national repository for incident end-stage renal disease, and you can just see how much greater the incidence of end-stage renal disease is in African Americans or blacks. And it's also high for other minority groups as well. If you look at so they've been recording data since about the 1980s, and if you look at the incidence over, the last few decades, you can see that the incidence for African Americans has been higher. It's decreased for Native Americans, and has been sort of steadily increasing for other groups. And that decrease in Native Americans really came about when we started to treat the diabetes that was sort of rampant in some of the Native American communities with all the things that we know that we can treat. So blood pressure was treated, diabetes was better controlled, and people had access to nutritionists, and you can see that the incidence actually declined. So we know the things that we need to do to really decrease the incidence of end-stage renal disease, and I think it's really making sure that people have access to health care so that they can get the care they need to actually decrease end-stage kidney disease. So a little bit more data about the sort of lifetime incidence of end-stage kidney disease for men by race and ethnicity, and you can see that in our country, end-stage kidney disease starts about 10 years earlier for African American men compared to others, and it's the same for women. So why is there this discrepancy? Why do we think this happens? So a lot of what we think contributes to that difference are determinants of health and kidney disease. So the social determinants of health, of which you've probably heard a lot about, include things like education, healthcare access and quality, neighborhood and the built environment or what people are exposed to, social and community contexts, and economic stability. So it's that range of personal, social, and economic and environmental factors that influence health status, which are considered the social determinants of health. So this is a conceptual model that Yoshio Hall, who's a nephrologist, put together to think about how changes in the social determinants of health can affect the development of kidney disease. So if you start at a certain socioeconomic position, that can affect your social class. It can affect what education you have, what occupation you get, what your income is, which then can affect your access to the healthcare system, whether or not you actually have insurance. That can affect lots of other material conditions. Do you have food, housing? That can lead to different biological factors and psychosocial factors. So all of that can contribute to the development of hypertension and diabetes, which are some of the biggest risk factors for kidney disease. So my research is really just focused on looking at risk factors for kidney disease in the African American population. These are some of the typical risk factors for kidney disease, and they include hypertension, diabetes, a family history of kidney disease, smoking, poverty, and lack of access to care, cardiovascular disease, and then some genetic diseases that are associated with kidney disease. So my research is really focused in on the African American population, and to do that I actually got a grant to look at the Jackson Heart Study, which is one of the largest prospective NIH-funded trials of cardiovascular disease in African Americans. It involved about 5,306 African Americans from Jackson, Mississippi, and they started the exams in the year 2000 and have now been funded to do another exam that started in 2021. And they've been following this group, really looking at outcomes for cardiovascular disease. And so we were funded to look at kidney disease, and so that's what I'll share with you today. So one of the first things that we did was to look at risk factors for rapid kidney function decline amongst a group of African Americans from the Jackson Heart Study. And just to give you an idea of what the cohort looks like, so this is all in Jackson, Mississippi. So there's both rural and urban populations. If you look at the whole cohort, it's a younger cohort compared to some of the other cohorts that people study. It's a higher percentage of women. There's a higher income for the whole cohort, and education, those who have less than high school education only make up about 20%. Many have private insurance, and blood pressure is controlled, creatinine is normal, and about 22% have diabetes, and about 61% have hypertension. So, and the majority of people who were enrolled in the trial did not have kidney disease to start with. So, so when we looked at risk factors for rapid kidney decline, which was defined as an eGFR decline over 30% over 10 years, what we found is that the higher the increase in age increased the risk of kidney disease. So 1 means that there's no risk, less than 1 means there's protection, and greater than 1 means that there's an increased risk. So low income was associated with a twofold greater risk of rapid decline of kidney function. Lower education was associated with decline in kidney function, higher blood pressure, history of diabetes, smoking, and history of cardiovascular disease. So many of these are social determinants of health, so that wasn't surprising, but this was all within this population that are self-reported as African American. So we concluded that some chronic kidney disease was prevalent. It was about 11% of the population at the beginning. The risk factors for rapid kidney function decline included socioeconomic status, which included income and education, age, diabetes, hypertension, and cardiovascular disease, and smoking, which was new. BMI and waist circumference did not appear to be risk factors for kidney disease. So one of the cardiologists was interested in smoking. So I wanted it was the first time that we had found that smoking was associated with kidney disease, so we went through and looked at how was smoking associated with kidney disease decline. And so we looked at current smokers versus past smokers versus never smokers, and found that if people were current smokers, they had a more rapid decline in their eGFR at -12 compared to past smokers compared to never smokers. And if you looked at those who never smoked and looked at them and said that they were, sort of the referent group, past smokers had an increased risk of rapid kidney function decline as did current smokers, and it really depended on the number of cigarettes that people actually smoked, so which was very interesting. Another investigator, Casey Rebholz, was interested in beverage patterns and whether that influenced the incidence of kidney disease. So she looked at the cohort, included those who she had information for what they drank, and created a evaluation where she adjusted for lots of different things, including total energy, their age, their sex, education, BMI, smoking, a lot of these things that we know are associated with kidney disease. And what she found is that if people drank or said that they drank a lot of soda or sweetened fruit beverages, that they had a higher incidence of kidney disease. So looking at things that people can actually modify could affect whether or not someone actually develops kidney disease. So we went through and looked at a lot of other things that I took out of my talk because I wanted to make sure I got to APOL1. So some of the other things that we found were associated with kidney disease that were new, that cardiovascular disease, if folks came into the study and had evidence of cardiovascular disease, that was definitely associated with increased risk of kidney disease. The other thing that we found as a group is that sickle cell trait was also associated with increased risk of kidney disease. So that I'm going to switch to talking about genetic diseases that are associated with kidney disease. And I bring sickle cell trait up because in the past, many people thought that sickle cell trait was not associated with other diseases. And so we pulled information from lots of different cohort studies and showed that that was the case. So I'm going to tell you a little bit about APOL1 and start the story and talk about how this sort of developed and then talk about whether or not we should be thinking about doing genetic testing in the African American community. So in 2010, this paper was published called The Association of Trypanolytic APOL1 Variants with Kidney Disease in African Americans. And this is one of the first papers that showed that variants in APOL1 were associated with increased risk of kidney disease. And so trypanosomes are parasites which are endemic in Africa. They're transferred from the gut of the tsetse fly to humans by a bite, and that's a trypanosome there in blue, and those are red blood cells in red. They live inside the human body and they can multiply rapidly. And when they cross the blood-brain barrier, they can cause African sleeping sickness. And so that's one of the methods of African sleeping sickness. And it is thought that APOL1 and some of the genetic variants actually protect people from trypanosomes and protect them from developing African sleeping sickness. So these variants are thought to have arisen some 3,000 to 6,000 years ago in Africa, and they're associated with resistance to the lethal form of African sleeping sickness that people can get from the bite of a tsetse fly. These variants are only present in people of African descent from a certain geographic area, and someone is thought to be high risk having kidney failure if they have two of these risk variants. And G1, so the two variants are G1 and G2, and G1 has two amino acid substitutions that you can see there. G2 has two amino acid base pair deletions that you can see there. G1 is present in about 40% of the Yoruba tribe from Nigeria and West Africa in a cohort called the HAPMAP, where they had participants from Europe, Japan, and China, as well as West Africa. And it is thought that about 30% of African Americans have either G1 or G2 or both, and about 13% of African Americans have both G1 and G2 and are considered high risk for APOL1 variants. It's thought that this is an autosomal recessive disorder, or that it's passed on as an autosomal recessive gene, and is what has been associated with kidney disease. So the APOL1 or apolipoprotein L1 is something that actually can protect people from these trypanosomes because it blocks the lysis. Trypanosomes have created a defense called the serum-resistant active protein that can bind to apolipoprotein L1, and it inhibits it from basically going into the membrane, and then inhibits lysosomal swelling, which causes the parasite to die. So if someone has a G1 or G2 variant, then that sort of blocks the serum resistance activity protein, and then the trypanosome basically dies. So so one group of investigators looked at G1 and G2 and the wild type in Africa, and what they saw is that there was a higher population of people who had either G1 or G2 variants in West Africa, and those who had the wild type or just the regular apolipoprotein were more likely to be in East Africa. So again, sort of looks at this geographic sort of isolation of West Africa, sort of similar to what we think is happening with sickle cell disease. And so if people have like none of the high-risk phenotypes, then they are actually can get trypanosomiasis. If they have one of the risk alleles, they have an advantage. And if they have both alleles, then they have more of an advantage. But if they have both alleles, then they're more likely to either get what people think is something called APOL1 nephropathy. And so since that paper was published, there have been lots of papers looking at the increased risk of end-stage kidney disease associated with APOL1. So there was a paper that looked at focal segmental glomerulosclerosis, which is usually associated with hypertension, and found that of those folks who were assessed, there was a 10-fold greater risk of end-stage kidney disease, primarily in African Americans. For hypertensive-associated kidney disease, there was thought to be a 7.3-fold greater risk of end-stage kidney disease. And then for HIV-associated nephropathy, there was a 29-fold greater risk of end-stage kidney disease. There have been several studies done in transplantation that show that there's an increased risk of progression of kidney disease and development of kidney failure in those who, if someone gets a kidney from a cadaveric person who donates a kidney. There have been a few other studies that have looked at APOL1 high-risk genotypes and just the risk of kidney disease over time. And if people have 0 copy or one of the copies, then their risk is basically the same as shown here. But if they have 2 copies, then they have a greater risk of end-stage kidney disease development. So this is just a study, or shows all of the different studies that have been done. So the initial studies were case control studies where they would pull out cases and look at controls, and they had these very high odds ratios. But when they looked at chronic kidney disease cohorts or population-based studies, the odds ratios or hazard ratios decreased because you're looking at the whole population and not just the people who had a case of kidney failure. So we really think that if someone has a high-risk G1 or G2 allele, they're probably in this area where they have a 2-fold greater risk of end-stage kidney disease and kidney disease progression. So this is another study that just shows kidney survival in those who have APOL1 who have FSGS, and it just shows again that if people have the 2 risk alleles, their kidney survival is less than those who have 0 or 1 risk alleles. And this was a study done by Carmen Peralta down at UCSF, where she looked at the Cardia study, which is a study of young adults, or they were young at the time that they were enrolled to the study, and looked at the onset of albuminuria, which is one of the first signs of kidney disease, and the risk of kidney failure or decline in eGFR. And what she showed is that those who were in the high-risk APOL1 group, they had an earlier onset of albuminuria, and then they had a much faster decline of their eGFR compared to the white group or the low-risk black group. The study participants who had the low-risk genotypes. And this is, again, one more study that shows that APOL1 variants are associated with a higher risk of transplant failure if someone receives a kidney from a cadaveric donor who has APOL1, who has a high-risk genotype. So we looked at APOL1 and albuminuria and dialysis and CKD and kidney function decline in the Jackson Heart study because as I was concerned that if I had one allele, then I might be at a greater risk for kidney failure. So what we found is that we probably didn't have enough power, but if people had 2 of the high-risk alleles, they had a almost 2-fold greater risk of early kidney disease. They had a 9-fold greater risk of going on dialysis, a 1.65-fold greater incidence of chronic kidney disease, and a 2-fold greater risk of eGFR decline. So in this population of African Americans, we see that this genetic finding is associated with kidney disease and kidney failure. So that started the journey for me of thinking about genetic testing. So as these results started to come in, many more people thought, well, we should be testing everyone for African Americans for APOL1, like we have done for many other diseases. And as we started to think about genetic testing, what came to mind was this, and I don't know if people recognize this, and I always ask the audience, and someone can say if they recognize it. It's Tuskegee. So, and I show this because there's a legacy of mistrust with the medical profession that has somewhat to do with Tuskegee. It also has to do with sickle cell testing of newborns, programs in the 1970s that were associated with mistrust, where people were, and actually there were programs where people were tested for sickle cell disease before they actually were allowed to get a job, or they were tested for insurance purposes. So there's a lot of mistrust in the Black community regarding this. There's a lot of mistrust in the Black community that has to do with a lot of what goes on in medical care, and that's just part of it. And the other part of genetic testing is that if we don't have anything that we can offer people, or if it changes their ability to get health insurance, then as a nephrologist, we have basically had a policy of don't ask, don't tell. So sometimes we'll actually make a diagnosis of something like polycystic kidney disease. And the parents who are in their 40s or 50s say, well, I want my kids to get tested, and before Obamacare, it's like, well, then it will change their ability to get insurance. So maybe you shouldn't, or you should only test unless you have a very specific reason to test. So there are a lot of questions regarding testing, and part of the questions arise as to what is the right way to start genetic testing? Should everybody be screened? Who has access to the information? Do insurance companies have access? How long is that information kept? What if we find out that a disease is associated with genetic abnormalities that were thought to be benign when we actually did that testing, like sickle cell trait? And do people have a right to know if they were screened for disease as a child, and now it's been found to be associated with an actionable disease? And as researchers, we do a lot of research and are doing a lot of genetic discovery, and a lot of that information is not given back to patients. So I worked with Wiley Burke, who's a geneticist. Now she's retired. Malia Fullerton, Erica Blackshear, Clarence Spigner, Jonathan Himmelfarb, where we wrote a grant to look at community-based evaluations of genetic testing in African Americans. And we wrote this grant, got it as an R01, really to conduct interviews to determine the views about APOL1 status for research participants, for patients, for family members, and for the community. We conducted community-based discussion groups or town halls to identify community preferences and priorities for APOL1 testing. And then we convened a national meeting of stakeholders to really review all the evidence that we had found with the community conversations that we had to help us develop guidelines for policymakers. So we did this process, and I'll walk through the process a little bit more, but we started with 3 different groups of African American community members, self-identified. We did interviews, and then we had deliberations. We had this national meeting, and then we made these, came up with recommendations, which included developing educational materials, no routine testing, kidney transplant testing, and offering participants APOL1 status. So we started with key informant interviews back in 2016 to 2017. We had community conversations that took about a year, and then we planned and had this stakeholder meeting to really develop policies and guidelines on educational material. The study took place in Seattle and Nashville and Jackson, Mississippi, and that was just because we had folks who could work with us in those different places. We did about 75 interviews of researchers, of clinicians, which included nephrologists and primary care providers, and also of community members, which included patients, family members, and other community members, and asked lots of different questions. But one of the questions we asked is, should we offer APOL1 testing to all patients, irrespective of other risk factors? And what we found is that the researchers said, no, we should not offer testing because we don't know what we're going to do with that information. Nephrologists said, most of them said, no, we shouldn't offer testing. Primary care providers thought, well, maybe we should offer testing. But the patients, you can see, really thought we should offer testing, and family members and others. And when we had the conversations with the communities, we were getting questions like, why haven't we heard about this before? You've known about this for over 10 years, and this is the first time that we're hearing about it. And we want to know that information so that we can make those decisions for ourselves. What the community conversations, what we heard was that there was a lot of support for optional APOL1 testing in the routine patient care and kidney transplant. And what one person said is, we've got to know, first thing I would do would be to live a healthier life, change my lifestyle, and maybe they might find a cure at some point or a way to help us. Other participants expressed really strong support for APOL1 in the transplant setting. And there was near unanimous support for offering testing to potential living donors. But most participants really opposed a policy of prohibiting living donors with high APOL1 risk from donating a kidney. And so we brought community members together with researchers. Martin Pollock, who actually it was in his lab where they found APOL1 for this advisory board. And we presented information, we presented the research from the community deliberations. We had representation from the NIH and NHGRI, also from some of the policymakers and from foundations and from the genetic organizations. And it was at this meeting where the researchers were actually meeting with patients who had kidney disease, where one of the researchers was actually crying because he just realized that his research had such an impact and made such a difference for patients. So it was very powerful. And conclusions from that meeting were that African Americans should be informed about APOL1 risk. APOL1 testing should be integrated into renal transplant programs. APOL1 testing is not recommended for routine clinical use or screening because there are no specific actions or treatments to improve outcomes for people with APOL1. And research is needed to better ensure understanding of APOL1 risk, and involvement of members of the African American community in development of policies and educational material about APOL1 risk and APOL1 testing are important, so that the community's needs and preferences are addressed. So I'll go back to my case with Mrs. J, who underwent this gastric bypass, and then she received a kidney transplant from her 18-year-old daughter. And what factors are important in thinking about her transplantation. So one thing that we couldn't test for back then was whether or not her daughter had APOL1, because should her daughter, who was 18 years old and able to make a decision on her own, be allowed to give a kidney? And at the time, and I'm not sure it's still going on, that APOL1 came out, many of the transplant centers were actually testing for APOL1 and then telling living-related donors that that they should not give a kidney. And what we heard from the community is that the community wants to make that decision. People want to be able, if I'm 65 years old and I have 2 kidneys that work well, I want to be able to give a kidney to someone who's younger. But that's one of the questions. And would this change your decision to offer a kidney transplant? And then, are there new biologics available for treatment of APOL1? Because if there are, then we now have a treatment that's available. So I put this in because this is pathology and laboratory medicine, and there are papers out there that do show that there might be an APOL1 associated nephrology, and it's associated with this type of histology. And many nephrologists don't necessarily biopsy certain people who have hypertension or diabetes because we think that we know what we're going to see. And so this, I think, might be a reason for us to do more kidney biopsies. And there's the kidney KPMP, or Kidney Precision Medicine program, where they're actually doing biopsies, and we'll figure out what some of the biopsy findings are, and will give us a lot more information. So this is what APOL1 associated nephropathy is thought to look like. You have these solid glomerular structures, you have interstitial fibrosis that's there, thyroidization of the tubules, which I think that's there, and some microcystic tubular dilatation, which is there. And there are new tests for APOL1 detection. So Andy Hoofnagel, who I think couldn't come, but published this paper and has this project that, so if anyone is interested, he has this project that needs to be completed where he's actually looking at APOL1 protein in plasma, and it might be able to be used to really do a test that's not a genetic test, but very specific for APOL1. And I put this slide in just to show that there are new drugs that are targeting where people think APOL1 is having an effect. So Vertex, there's a drug by Vertex, by Ionis, and they're in the process of testing these drugs. So a few minutes for discussion. Racial and ethnic health disparities in kidney disease exist. We know that social determinants of health and other risk factors can really affect the development of kidney disease. APOL1 is a genetic variation that's associated with kidney disease, and studies determining the ethics around genetic testing for APOL1 are ongoing. So I want to say thank you and stop for questions, and also want a lot of gratitude for the Jackson Heart Study CKD working group that includes several researchers across the country, and funding and support for the research here. And stop for questions.
Betsy, hi. Long time no see. Thank you very much. Really, really nice talk. So I guess I was kind of going to ask, but you may have answered in your last slide. I was going to ask, so if you really can't, if there are no interventions that are available based on APOL1, I guess why test for it?
Right, and that is what it came out during a lot of the conversations. If we don't have an intervention, why test for it? But now there are these biologics that are probably going to be something that is going to be tested now in clinical trials. What I will say is what the community has said, that you may not have something that you can treat me with, but I would want to know if I have APOL1, because then I might want to make sure that my children know. And people will say, we're supposed to be doing all these things like controlling our blood pressure and treating our diabetes, and maybe that might make me be extra careful about those things.
I guess one other question, if I may. You mentioned sickle trait briefly. Would you care to elaborate sort of on some of? I always associate sickle trait with when you get older, you sort of lose the ability to concentrate urine. That's kind of the main thing that I know about. Are there other things that sickle trait?
Yeah, so I took those slides out because I thought I was going to go over, but so we did a study with the Jackson Heart study with Mesa, with I think Eric, where we pooled all of the African American population. So there's a population of, and the question was, what about sickle cell trait? Sorry, I'm repeating the question. So we did a study where we pulled all of the African Americans from all those different studies. There were about 16,000 African Americans, and we looked at whether or not those who had sickle cell trait developed kidney disease, or have developed albuminuria, or developed faster progression of kidney disease, and they did all of those things. And so that was new because as you said, what we know as like nephrologists is that maybe as people get older, they have some ischemia in the kidney, and it's associated with all of these things that are associated with ischemia, but there's probably something else that's going on. What the mechanism is, I can't tell you, but I suspect it's part of that, but definitely there are those associations with albuminuria, development of chronic kidney disease, and faster progression of kidney disease as well. Jim?
Thank you, wonderful talk. My question is about whether or not there is synergy between the risk imparted by APOL1 and some of the other risk factors that you show. So are sugary sodas worse in APOL1 than not? Because that's the basis of maybe an intervention like you were talking about.
So I'll try to repeat the question. Is there synergy between some of the known risk factors for kidney disease and APOL1? And there probably is, and I think that research needs to be done, but we know for hypertension there is, and if you treat hypertension, it actually improves, there's a benefit for those who have APOL1. Sugary drinks, I don't know if anyone's actually done that study or some of the other things. People have looked at diabetes, and it appears that diabetes doesn't have an effect, but I suspect that many people have both diabetes and may have APOL1, nephropathy, if that's an entity. And if they are faster progressors, it's probably the APOL1 that's doing it as opposed to the diabetes. But that's a great question.
I just wanted to add something to the conversation about whether or not to.
And maybe you should use the mic, because then I have to repeat what you say.
Okay.
Come up.
Okay. So I just wanted to add something to the conversation about whether or not there should be testing for patients. And one of the other things that should be thought about is the second hit hypothesis for APOL1, right? And so Andy, in his paper with the serum, used sort of a second hit on the kidneys. And so he expressed APOL1 variants in the liver to create circulating APOL1, so not directly from the kidney. But then they did a second hit, a drug hit, on the kidney. And so kidneys that had the circulating G0 did not develop nephropathy. But kidneys that had that second drug hit but had the circulating G1 and G2, I think G1 in particular, did have nephropathy. And so I think there is a reason to test patients, because I think that there's ignorance about APOL1 in the patient population, but I think, and I say this as a non-physician, I think there's ignorance about APOL1 in the physician population as well. And so your family doc may not know anything about the fact that you potentially have kidneys that are more susceptible to drug damage, for example.
Thank you.
Here's a question from Melissa Upton online. In your role as DEI Dean, can you address how UW will lean back against national backlash against teaching critical race theory and other concepts essential for understanding marginalization, inequities, and gaps in healthcare justice and access?
That is a great question, so I'll try to repeat it. So in my role as the Vice Dean for the Office of Healthcare Equity, what are we doing to sort of lead the way against the backlash against a lot of the diversity training, critical race theory, all of those things that our office is really doing. And I would say that we will continue to do that work, because we have to. We're in the process of becoming an anti-racist institution. And in order to do that, we are promoting that people need to start on their equity, diversity, and inclusion journey. And in order to do that, you have to have education about it, which is why we're offering foundational training in 5 different areas. And that it is a process, right? We all come with our own biases. We all have our whole history of how we grew up. But when it comes to our patients, we have to make sure that we're treating our patients well, and that our biases don't influence how we treat our patients. And that's what our office is really, that drives our office. That we have to treat each other well. We need to make sure that we're treating our patients well. And the only way that we're going to get there is by educating people.
More online if there's not more live. So 2 anonymous attendees ask, will APOA1 testing of kidney donors potentially decrease the number of HLA-matched donors of African ancestry? And then, do you know if 23andMe and other social genetic testing platforms are testing for APOL1, and should they test?
So I may ask you to repeat one of the questions, but the one question is about APOL1 and transplanted kidneys. So there is an ongoing study called APOLO, where they are actually monitoring and testing for APOL1, and they will be testing living donors and looking at progression. So some of those questions we don't know. I think right now There are no regulations about whether or not we test for APOL1 and whether or not somebody is told that they have a kidney that is from an APOL1 donor, which is a lot of what we brought up in our discussions. And so it's up to the transplant center if they want to test for it, if they want to tell the patient about it, if they want to have the living donor know what their status is. And so there are a lot of questions there because probably having a kidney that lasts maybe for 5 years is better than having a kidney than having no kidney at all because of the high mortality rate associated with dialysis. And then the second question?
Do you know if 23andMe and other social genetic testing platforms are testing for it, and should they?
So I don't think they test for it yet. I know one of the researchers who's a transplant nephrologist, Barry Friedman, was very interested in having that done. I am not sure, but I know that a few years ago that they had not been starting to do.
I just looked on my phone because I was thinking the same thing, and 23andMe does offer.
They do. They do offer APOL1. You have to pay extra for it. And so the question would be, do they offer you educational training? Like I think they offer BRCA1 testing as well. So, well, there you have it. And there's another question in the back.
Have you found APOL1 in other races apart from African Americans? And when we test for APOL1 in the donors, do we just test the African Americans or we test other donors as well?
So that is a great question, which gets at what is, African American, what is race. So a lot of the studies were done in populations that were either white or people self-identified as Black or African American. So if you have African ancestry, you may be at a higher risk of having an APOL1 gene. So as you probably know from what you've heard about Ancestry.me and a lot of other ancestry tests, many people don't know that they have African ancestry. So to me that would mean if someone has kidney disease or is thinking about it or concerned about it, then they probably should be tested for it.
Sue Quagan published a paper with a white patient with the APOL1.
Yeah, and it's the same for sickle cell, sickle cell trait. At our conference there was a white researcher who knew that she had sickle cell trait and but didn't know that she had any sort of Mediterranean ancestry. So it was a shock to her and started her down this whole pathway. Question?
This is kind of a follow-up to that. So have they identified any APOL1 variants in other geographic locations that have the trypanosomes present, so like South America or,
I don't think so. And it really is sort of that geographic area because if you go, and I don't know if there's mountains or what the geographic divide is, but if you go to East Africa, most people who are tested do not have APOL1.
Different tsetse flies.
Different tsetse flies. So
Brad Cookson asks a question similar to that. So you drew these comparisons between sickle cell and malaria. Do you know if APOL1 mutations contribute to anemia independently of causing kidney disease?
I don't think so, but that's a great question. So the question was, does APOL1 genotype, does that contribute to anemia like sickle cell disease and trait do? And I don't know the answer to that, but that's a great question that we could answer in some of the databases that we have. And question up high.
I just was wondering, were any insurance companies or I guess, representatives of insurance kind of involved in the community evaluation process just to think, we could assume that they would probably be predatory on the information, but I'm just curious.
So we did, at that meeting, we did not have insurance companies there. We did have the geneticists there who told us, who had the experience of doing genetic testing for other diseases. And they said that if you did testing, it would, be billions of dollars to do that, if you started like doing testing at birth. And I think it's a great question because once insurance companies know that someone has APOL1, then, they're the best biostatisticians. They know who's going to develop disease and they will charge you for it or not allow you to get health insurance for it. Thank you.